episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you not one, but two fascinating guests working in some areas, creating a better tomorrow for all of us in the process. If you've been paying any attention to the really cool non-COVID scientific literature over the last couple of years, you may have come across the term Xenobot in the press. These living micro robots that are built from cells have been demonstrated in the laboratory to do all sorts of cool things in terms of moving towards targets, picking up payloads, healing themselves, recently being able to show to reproduce via a fascinating process called Kinematic Selfreplication. And in addition to all the futuristic potential that has been mentioned in the press about cleaning up radioactive waste, collecting microplastics, maybe one day helping us terraform planets, they are additionally offering this fascinating new toolkit for helping us understand complex processes such as body morphogenesis, how complex tissues form, ultimately how minds develop, and even give us glimpses of possibilities of what novel life forms we may encounter one day out in the cosmos. And this cutting edge research has been conducted by an interdisciplinary team composed of scientists from both the University of Vermont, Tufts, and Harvard. And our show is honored to be joined by a couple of them today. So first, joining us again from a recent episode, Dr. Mike Levin, Tufts University professor who holds the Vannevar Bush Endowed Chair in the Biology Department, serves as both the director of the Tufts Center for Regenerative and Developmental Biology and director of the Allen Discovery Center at Tufts. Mike Levin, welcome to our show.
Thanks so much. Really happy to be here. Thank you.
And also, a friend of the show, Dr. Josh Bongard, professor of Morphology, Evolution, and Cognition Laboratory, Department of Computer Science, College of Engineering and Mathematical Sciences, University of Vermont. Dr. Bongard's lab focuses on the really unique domain of evolutionary robotics with the goal of ultimately directing evolution of increasingly complex, capable, and autonomous machines to perform a wide range of tasks. Josh Bongard, welcome to the show.
Thank you, Ira. Thanks for having me back on.
Absolutely. And while we are introducing you two, we have to mention two other folks on the team that are not with us today, but nonetheless, Dr. Sam Kriegman from Harvard University, who did his PhD at University of Vermont under Dr. Bongard, as well as Dr. Doug Blackiston, Senior Scientist at the Allen Discovery Center at Tufts, with Mike, who has been focusing a lot on understanding tissue plasticity. Really fascinating set of themes here, and really interdisciplinary team. Well, anyway, that being said, welcome, gentlemen, back to the show. And I'd love to start off just with a basic question. Obviously, somewhere between you guys getting together at a bar and sketching this thing out on a napkin, or one of you having a Peter Parker Spider-Man style accident where the Xenopus embryo started hopping around the lab, the idea for this came to fruition. Take a little time, both of you, to just talk about the early days on when this idea first materialized between your group.
Well, I guess the biology side can be traced back to really fundamental questions about novelty and autonomy. So standard embryonic development kind of lulls you into a false sense of security because it's so reliable. And we watch these things form, and we kind of get the idea that, okay, fish cells make fish and acorns make oak trees and so on. And you get this idea that that's the way the process has to be. And for a long time, I, and of course other people have thought about this as well, really have been interested from an engineering perspective in understanding what else is possible. So given those same exact cells, that same genome, how do the cells know what to do? Because what the genome gives you is actually the micro hardware of the cells. It doesn't actually describe anything directly about the shape or the functionality of the creature. So what else is that hardware capable of? And so people in the past have thought about, of course, epigenetics in terms of phenotypic variability, how the same genome might look different, let's say, especially in plants under different circumstances, and chimeras. So people, in fact, we recently published a review of chimerism going all the way back really classical data, looking at putting cells together that don't really belong together, and how they, what are they going to do? So this idea, and so we were thinking for the longest time about what is a really good tractable model system in which to understand why do cells do the things they do in collectives, and then what else are they capable of. And so I've been thinking about this for a long time, but one specific aha moment was actually in a PhD thesis committee meeting where Kelly McLaughlin, who's a colleague of mine in the biology department, her student Emily was showing some work with pieces of Xenopus and of Xenopus embryos, and we noticed that they were sort of running around on their own in the dish, aside from anything else that they were trying to do. If you sort of look carefully, it's like, wait a minute, they're moving around, and I sort of immediately thought that, well, this might be a very sort of minimal biorobotics platform. And after that, we had some conversations. I had conversations with Doug Blackiston, who then immediately had the idea of letting them reconstitute themselves from individual cells. So rather than cutting chunks, which we've done, but just dissociating cells and letting those cells reassemble from scratch. And then we had some great conversations with Josh and Sam that Josh will tell you about how we can actually leverage all of the advantages of AI and machine learning to understand this very complex process that humans have been trying to understand for many decades, and frankly we need a lot of help with it. So
Josh?
And so from the robotics point of view, I've been working in this space for a number of years, and I've really been guided by this idea of embodied cognition, that the body scaffolds the development of cognition. And it's been hard going. We're kind of rowing against mainstream thought in AI, which is that brains equal intelligence. This is a very, very old idea in Western thought going back centuries. So it's often an uphill battle trying to demonstrate and argue and show that, bodies matter as well. For a number of years before Mike and I teamed up, we tried to do some demonstrations with rigid robots. We pulled off the leg of a robot and wanted to show how it could recover behavior with this changed body. In the last 6-8 years, we've been working in the space of soft robotics. This is a really exciting space in robotics where we're trying to make intelligent machines out of increasingly exotic, non-biological materials. So once we teamed up with Mike and realized that Mike and Doug could start to create sculptures and then machines out of biological tissues, we went from rigid robots to soft robots to what are now known as biological robots or Xenobots. So it's kind of a natural progression that way.
Awesome. And continuing upon those two themes, so I think of Xenobots, and I talk to my friends or my kids about what you guys are up to. Here we have this novel life form, really, as you were saying, created at the intersection, on one hand, of biology, where we have sort of the Xenopus, this wonderful sort of workhorse of the developmental biology world for the last century or so, wonderful frog species that, aside from use in developmental biology, applications in cloning, the original pregnancy test based on it, all sorts of neat things happening nowadays in terms of reprogramming. On the other side, computer science, terms like evolutionary algorithms, artificial ontogeny, being able to do things with these artificial intelligence systems, different programs to practice, these possibilities. Talk a little bit and introduce us to sort of how the biologic and the computer components of this amazing research come together. Who does what first, when, and how?
Sure, I can talk to this from the computational point of view. So again, sort of looking at the history of AI and robotics, it's always been inspired by biology, right? We look around us, we are surrounded by fantastically intelligent machines, at least most of the time, built from biological components. And yet we have a surprisingly difficult time making intelligent machines out of metals and ceramics and plastics. So you mentioned evolutionary robotics. This is a very small niche inside of the overall robotics landscape, where instead of us reaching in with our hypotheses and our ideas about how intelligence should be embodied, we try and take a step back as designers and instead create what's known as an evolutionary algorithm. It's a computer program that runs inside of a supercomputer. And as the name implies, it tries to evolve machines rather than design them directly. It generates millions or trillions of virtual bots inside a virtual world. And it decides who gets killed off and who's allowed to make randomly modified copies of themselves. So this evolutionary algorithm is actually, screamingly simple. You can boil it down to about 10 lines of code if you know what you're doing. The virtual environment and building in all the biological detail into these simulated worlds, that's another matter. But that's sort of the workhorse on the AI side. Then comes the next challenge, which is once the evolutionary algorithm dreams up a way of rearranging virtual frog tissue to produce a virtual Xenobot that the evolutionary algorithm thinks might work in reality, we send that blueprint to Mike and Doug, and then the challenge becomes how to turn it into something real.
And Mike, when you get that blueprint, then what happens at Tufts?
Well, up until now, how it's worked is that Doug Blackiston, who's an incredibly talented sort of microsurgeon in addition to everything else, he attempts to implement it with a kind of, again, for now, it's been a subtractive process akin to sculpting. So basically what you do is you take the early skin cells from the embryo, you let them self-assemble in a little sort of depression. They self-assemble into a roughly a spherical shape. And then you can go in and sort of subtract or cut out little pieces to end up with a resulting shape. So for example, in some of the earlier work, when it was a mix of skin and muscle cells, you might cut out some tissue to end up with 4 legs. It's sort of like an ottoman kind of a thing, and see if the muscles will make the legs actuate so that the thing can move forward. So Doug carries out this sort of subtractive kind of sculpting process. And then whatever is the result of that, then comes the process of characterizing its behavior. So you might have just one that you're tracking, or you might have a variety of them in a dish that can interact with each other. They'll be swimming around. They'll spend some time connecting to each other, going off on their own, circling around the dish, all these kinds of things. And we have to record all of that. We have to quantify that. Maybe they're interacting with various objects that we place in the dish. We've had them in mazes, we've had them in tubes and all sorts of things. So then there's the process of characterization, and then there's the really important loop back. So then after we see what they do, then we have to come back and sit down with with Josh and Sam and say, okay, this is what we observed. What does this mean for the next phase of the modeling, right? What is this telling us, and what can we do, and how do we enrich the machine learning aspect to then make use of this information for next time? And it's sort of this like spiral that keeps going.
I think the feedback loop that Mike was just mentioning, this is where things really start to get interesting, because the AI and the evolutionary algorithm and the supercomputer, it's trying to build these designs, but it has incomplete information. We try and give it what we know about the biology of Xenopus in this particular case, but there's obviously still a lot of gaps. So the AI has to kind of, again, work with a lot of this uncertainty. When the AI suggests something and the Levin lab builds it in reality and then gives us back information about what worked, what didn't work, or a time lapse of what this Xenobot did or didn't do in the dish, they're hidden in that data is potentially, new biological information, new discoveries to be made. So how do we take that information from reality and feed it back into the computer, the AI, not just to build a better Xenobot, but to build a better understanding of living tissues? I think that's one of the longer term implications of the work we've done so far.
And that segues perfectly into sort of the first talking point in terms of what this type of research could lead towards in terms of this toolkit and its understanding. And you're just touching, Josh, on such an important one. Mike obviously published extensively about this, but form, morphogenesis, how that little cell that we start with becomes trillions, and how it decides to be arm, leg, eyeball, spinal cord, the shape and structure of something distinguished from what it's composed of. Mike, talk a little bit, if you would, about how these tools can, once again, as you do your work and air some of these unique biophysical studies on membrane potentials and mechanical transduction forces and so forth, help inform that work and take us down the path of sort of visions for what you learn in terms of regenerative medicine. And then Josh, at the same time, and here's the first of the childish questions you're going to get, but in this context of form, if you let your evolutionary algorithms run for X amount of time, at the end of the day, do you predict at some point you get Xenopus, a Xenopus at the end of the day in the computer? Do you get some weird Xenopus thing, or will you ever get a kangaroo or something else out of the evolutionary algorithm? Guys, take that in both directions, if you would.
Right. So I want to preface what I'm going to say by just a fair warning that this is not a mainstream opinion. Okay, so I'm gonna say what I think about these things, and I think some people may disagree with it. In fact, I'm sure I'll probably catch some flack from my biology colleagues, but I think that in an important sense, biology is it's a branch of computer science, and what I mean by this is that we have really fundamental, deep questions, both about the hardware of what's going on. And this is traditionally addressed with genetics, molecular biology, right? All these things that tell us what the pieces are, what the parts are that every cell gets to deploy. But then there's the software aspect, which is that once you have these highly active, competent little subunits that can do various things, meaning cells, the question of what is it that they're going to do in various circumstances is massively underdetermined by knowing what the hardware is. And of course, we know all this from computer science. It's nothing new there. It's important to realize what profound mysteries still remain, despite the fact that we have genome information and all of that, with respect to what's actually going to happen in various configurations. Because there are lots of examples, and I can go into these if you want and if we have time, where it becomes pretty clear that the results of the biological evolutionary process is not a solution that works well in a specific environment, right? But it's rather a machine that solves problems. And it's very good at certain things that evolution has already seen before and prepared it for, but it's also remarkably good at other novel circumstances that it may never have seen in evolution. And we have lots of examples of this amazing plasticity and problem solving in various spaces. This is not just behavioral intelligence, but this is intelligence in the space of anatomical features, in transcriptional or gene expression space, in physiological space. Living things are amazing at solving new problems. And this is not well understood at all. And so I think one of the biggest benefits of this technology that are going to appear, in addition to the useful stuff, right? The useful synthetic living machines that will be working in the body, outside of the body, and in factories, in the environment, doing all these things. In addition to all of that, the fundamental contribution of this kind of technology is going to be as a new kind of discovery engine to really focus our attention on important questions of where do goal states come from. And these might be anatomical goals, these might be behavioral goals, for a very simple reason. We're used to thinking that evolution is of course part of everything in biology. If you look at a certain organism and you ask the question, okay, why does it have a certain shape, a certain size, a certain color, a certain functionality? The typical answer leans on evolution. You say, well, because for millions of years, right, its ancestors were selected to do this and that. The systems that are synthetic, like these Xenobots that are, or chimeric systems, they are valuable because they don't let you have that easy out. There's never been selection to be a great Xenobot, right? And so insofar as Xenobots are able to do certain things and have certain reliable shapes, you have to now ask yourself, well, where do those come from? Because they weren't directly selected for. Now, of course, there have been other people that have sort of questioned the sufficiency of selection as the kind of the driving force of everything that we see. But this really puts it in a very stark and very experimentally accessible way. So to start to look at why do these collectives of cells, when they are basically, what we've done with them is, we haven't added anything. There are no transgenes. There are no nanomaterials, at least yet. There's what we've done is we've subtracted some stuff. So we've liberated them from the rest of the embryo, which means taking them out of a context in which the other cells are telling them to be a kind of external skin layer on top of these embryos and have this boring two-dimensional life. And when you remove those constraints, you find out that actually those cells are very ready to do something completely different. They're going to make a three-dimensional organism that's going to have a much more exciting life, moving around on its own and doing various interesting things. Where does that come from, and where was that? And really to start to understand development as a set of constraints that can be liberated to really see a lot of the creativity and problem solving that cellular collectives have. So that's what I think is really important, and not only for understanding evolution and embryogenesis, but really to help. And I'm sure Josh will talk more about this, but this idea that when we make Xenobots, it's not a traditional engineering process, because we are dealing with, and this is a new kind of way of thinking about it, that we'll see if people like this. I call this stuff programming with, or engineering with agential matter, in the sense that these are not dumb parts that you put together in exactly the right way. They're passive. You have to know everything about them. You have to know exactly how it's going to work. Each of the cells are themselves a competent agent with various homeostatic loops and things that it's trying to do. Yep. And making Xenobots are a collaboration. The cells do so much of the heavy lifting here, because we, let's say, free them from some influences, but a lot of the rest of it they do on their own. We don't know how to do any of that stuff, how to make them do anything. And so it's a collaboration. It's guided self-assembly. It's a new way of working with materials, where the materials themselves have all kinds of active processes as far as what they're going to do, right? And so it's we're trying to shape them and guide them. But it really is a kind of a two-directional collaborative process.
So I think these ideas that Mike is elaborating on, this throws down the gauntlet for AI and robotics. Mm-hmm. Especially when you look at AI, you hear a lot of language about optimization. You want to make a machine that does something better and better. So you have an end goal in mind. You know what that thing should be doing. You have a sense of, quote unquote, what it looks like, how it behaves. And the game in AI is to come up with a method that can sharpen or improve some base material or some base computation into that desired target. In robotics, you have a similar thing. You have bio-inspired robotics. You try and build a dog or a bird out of metal and plastics and electronics, because that's what you see around you. So you have, as a roboticist, whether you're aware of it or not, you tend to have, again, an end goal in mind, an end state. What happens if we now task the AI with working with a ball of potentiality, as Mike was mentioning? So it's silly putty, but it's not going to stay still. It's not the AI is playing with frog tissue and kind of pulling and pushing a frog into different shapes. It's moving through the space of what this potential material can become, which again is a very non-obvious thing for humans to think about. If I have an idea about what a machine should be doing in the real world, I can see it, and it's just a matter of building it from materials. How do we exploit, this ball of potentiality for whatever it is that we want to do? Ira, you also mentioned about, what might the evolutionary algorithm come up with, in future, which is a great question. I think my experience has been over the years, whatever you think it's going to produce, it'll be something else. So in our old work with traditional robots, we had this metal spider and we pulled off one of the legs and we knew exactly what would happen. It would stand up on the remaining legs and keep walking. That's absolutely not what it did. It lies down on its belly and kind of throws its body weight forward like the worm from the 1980s. In soft robotics, we continue to pull off the legs now of soft robots, and we knew exactly what a soft robot would do, which is because it can deform its shape, it's going to extrude the cut off leg and continue walking again. Mm-hmm. That's not what it does. It retracts the remaining three legs and again starts moving like a worm or rolling, which in retrospect is a perfectly viable solution. In morphospace, the space of all possible solutions to a given problem, there are so many good solutions out there that humans, we can't even conceive of until they appear. And that's part of what makes this so fun.
Absolutely. Josh, One thing I did not mention in your intro, I apologize, I'll bring up now. You, in addition to everything you do, you are also the co-author of two popular science books. One's entitled How the Body Shapes the Way We Think, A New View of Intelligence, co-author as well of Designing Intelligence, Why Brains Aren't Enough. And these books are about embodiment or challenging that, so intelligence requires a body. Mike, obviously, you've published quite a bit over the years in terms of all the organisms on this planet that do not have brains or formal nervous systems, of all those organisms whose brains you gently destroy, which grow back and somehow know things. Take us a little bit, both of you, take some time into the area of the mind now and what we can learn about how minds are formed, where intelligence comes from, what the minimal amount of material, in the case here of the Xenobot, we have no nervous tissue at all. This is muscle and skin. What are we learning in this research? What can we learn about the mind?
So you mentioned the book co-authored with Rolf Pfeiffer. Rolf Pfeiffer was my PhD advisor. Rolf Pfeiffer in turn worked with Rod Brooks, who used to be the former director of the AI lab at MIT, kind of the mecca for all things AI. And Rod Brooks, back in the 1980s, wrote a series of what became very influential papers. One of them was entitled Intelligence Without Reason, Intelligence Without Representation. And in those papers, Rod makes a point that Mike has also made, is that in nature, brains are a relatively recent invention. They're one of the last things that Mother Nature has come up with. Rod argued that if you look at AI, we're doing things the opposite way around. At that time, and still today with the deep learning revolution, we're focused on brains, brains, brains. So the way in which we're approaching intelligence, if we compare it to biological evolution, is exactly backwards. So what is it that was happening in that long evolutionary history before brains came about that we as roboticists are missing? What is that intelligence that's inherent or latent in the body that's useful? And there's been a bunch of work in this field. It's now known as morphological computation. Kind of an interesting idea that the body itself can compute. It can perform cogitation or computation that again supports intelligent thought. But it's still, at least for roboticists, a slippery concept. What exactly does this mean? How do I make a body that's smart or not so smart? What exactly does that mean? And I think that's where we really need to turn to some of these most recent discoveries in biology that Mike and others are making. That's really clarifying what exactly it is about non-neural tissue that supports intelligent action.
Yeah. One of the things that got me really kind of deeper into these questions was a challenge that was put out in it was a few years ago, maybe 4 or 5 years ago, at a Templeton meeting on diverse intelligence, and we were sort of pushed to try to develop a framework within which diverse intelligences could be compared. Now, when people think of diverse intelligences, a lot of times they think, okay, well, maybe, we're talking octopus, right? Or maybe we're talking, birds or something like this. But I started thinking about this question, I said, if we're going to make what is really fundamental about intelligence? Because I think the things that we normally take for granted, the size of the brain or look at the frontal cortex or whatever, these things, there's nothing magic in the material, I don't think. I think these are all contingent facts of the way evolution happened on Earth, but I don't think they are fundamentally what constrains or defines intelligence. And so what would it mean to be able to recognize intelligence in really diverse embodiments? So we're talking completely synthetic agents, software agents, things you might find on other planets, so exobiology, all kinds of chimeric sort of cyborg constructs, right, where we can now instrumentize brains and brain tissue and non-brain tissue with all sorts of electronics and other things, so to make these hybrid creations. And the framework that I've sort of been developing over the last few years is this idea that what is central about all of these kinds of agents is some degree of goal directedness, and the ability to, all the way from the most modest minimal things that are basically homeostatic loops around some sort of metabolism that all they do is allow you to persist over time, and that's it. But thinking about ways in which those kind of very simple homeostatic loops can scale up to larger and larger capacities that eventually end up with lots of reflexive, sort of the kinds of things, metacognition and this self-reflexive, hey, I know that I know, and these kinds of things that a lot of people think of when they think of cognition, they're thinking of sort of human level. And then frankly, past that, right? So it's pretty clear, I think, that there's no reason to think that we are sort of the pinnacle of this process. And I've been thinking about this idea of a cognitive light cone. So the idea is that what you can try to do is map out the size in both space and time of the largest goal that you as a system is capable of pursuing. So this is not the size at which you are able to sense or act. So this is not the range of your sensors and effectors. This is the size of the goal states that you are capable of working towards. So for example, if you're a bacterium, what you really care about is the local sugar concentration. And maybe you've got some memory going back 20 minutes, and maybe you have a little bit of anticipatory capacity, but basically it's a very small radius, both in terms of space and time, of the states that serve as goals to your homeostatic, as your homeostatic set points, basically. You can, but you can draw this kind of, and so if you imagine this sort of almost a space-time diagram where you collapse all of the space dimensions to one sort of horizontal dimension. You've got time on this axis, and so you can draw a little tiny circle, and that's the tick, the bacterium or a tick or something. That's the size of their goals, and anything bigger than that, they're just not going to be able to comprehend. And then you can imagine other creatures. You can imagine a dog, for example, that has considerable memory going backwards, some anticipatory potential going forwards. There's some spatial area it really cares about what happens. You are never going to convince that animal to care about what's going to happen 3 months from now in the next town over. It's just not going to happen. And so, right, and then you've got humans, which are actually capable. You've got people that are sort of working towards world peace and things that are going to happen long after they're dead. And the scale and the scope of the goals that motivate you to expend energy to do things in the world is a pretty good metric, along with another version of this is, which are the things that stress you, right? What are the states that can stress you out? And if you tell me what you're stressed by, I can pretty much guess your level of, of cognitive sophistication, right? Based on what's the most complicated the most complicated state you can be stressed by. So these kind of ideas are in many ways substrate independent. They don't say anything about what you're made of or how you got here. And I think that's really important, right? That whether you got here by natural evolution or by 2 steps removed, meaning you were made by human engineers who are themselves the products of evolution, that none of that is really super relevant. What matters is, what is your capacity to pursue counterfactual states that are not true right now, but could be if you were to work a little harder. They could be true. And so, what are you capable of representing? Given your makeup. And so that's the kind of thing we've been thinking about, with the idea that it should be a really kind of universal system that's very the goal of this whole framework, and I call it TAME, T-A-M-E, for technological approach to mind everywhere. The idea is that it should really be a very engineering approach. We shouldn't be making philosophical pronouncements about whether a given system does or doesn't have cognition. You should be able to do experiments, right? And you should be able to say, okay, what is the kind of what's the level of agency that I need to attribute to this thing to get proper or optimal behavior and control and prediction of what it's going to do. And and
outstanding, really outstanding. Good.
Yeah, those ideas actually resonate with some ideas that have been relatively vague, in my opinion, in AI for a while, which is a related concept to embodiment called empowerment. And this is related to kind of what Mike was mentioning, which is, what are all the ways you have at your disposal to keep your options open for the future? In the empowerment literature, it's usually about survival. How long into the future can you keep yourself going? I think Mike's approach is generalizing this idea to what can you make happen, in the future. So I think, again, there's some interesting points of synergy between some old ideas in AI and some new ideas in biology, and it'll be interesting to put those together.
Absolutely. So we've been discussing how Xenobot research can help us moving forward, looking at morphogenesis, thinking about minds in general. Want to segue now in the area of just life in general. And if bear with me for a second here, but people that listen and watch my show know that aside from I'm interested in doing this show, I'm a huge fan of science fiction, and one of my favorite films in the genre is the 1982 John Carpenter classic, The Thing, starring Kurt Russell, and my little poster over here. Kurt Russell was not at the comic book convention that day, but was signed by Peter Maloney, who plays one of the characters. But in the 1982 classic, the alien reproduces in a way, not like the Xenobot in the Alien series with eggs and gestating in a human host, but by a method that sounds interestingly and looks a lot like kinematic self-replication, where you could destroy most of the thing, but if there's some remaining, it could reproduce itself and keep going and having a fine day. Talk a little bit, guys, if you would, about what you've learned. And obviously, you published on this process a couple months ago. What are you learning about this unique movement, a form of reproduction, and what is that informing? And Mike and I, we've had the discussion on past episodes about what this may imply for when we think of astrobiology and some of the stuff that those folks think about in terms of what we may encounter out there in the cosmos one day. But talk a little bit about what you learned during this process of the research program and where you might be guided via some of these learnings.
Right. Well, I want to start by saying nobody's a bigger fan of The Thing than I am, but I think we need to say that. Well, maybe you, Ira, but I think we have to be clear that the Xenobots do not do what you saw in that movie, because the replication that we showed takes place under very, very special circumstances,
sure.
Right? So what they're doing is, what the bots are doing is that if provided with loose cells of their own type, they will run around and sort of collect them into little piles. And then this is super interesting. The Xenobots do exactly what we do when we make Xenobots, meaning that they lean on the ability of those cells to self-replicate. So the Xenobots don't micromanage this process any more than we micromanage the initial generation 0. They will do the first step, which is what we do, which is collect these cells into little piles, and then they let the piles take care of themselves. And what the piles do, of course, being in the case where the piles are themselves frog skin cells, they will make Xenobots, and then, of course, they will do the same thing to the next generation. So I find it very amusing that this is a kind of self-similar process where there's no micromanagement at any level, and it's a collaboration. We collaborate with the cells to make the bots. The bots will do the exact same thing with the next generation, and so on. And so all of this only happens when they are provided with these cells. That's, by themselves. They don't do the typical thing that a lot of biological organisms do, which is either bud or in some way, replicate, the grow and then divide or something like that. I think it's also very interesting about what's also very interesting about that process is, if we sort of, again, thinking about it from the point of view of novelty, surprise, and problem solving. Think of what we've done is we've taken some cells that have a genome that has been shaped to be really good at directing a particular kind of reproduction. It makes frogs, the frogs mate, the frogs lay tens of thousands of eggs, and you get all these tadpoles. I mean, it's an amazing process. We now come along and we make it impossible for these cells to participate in that process. We remove them from the embryo. They're just skin. They don't have any of the other stuff that you need to go through that cycle. And within 48 hours, they discover a completely novel way to get this done. Now, this is not the same as the true reproduction of a lineage. For example, they don't have strong heredity. So they don't have the kind of heredity and process that you would need for true evolution. Or it's a minimal model. It's exactly what you would expect from a minimal model. But nevertheless, I think it's amazing that once you've closed off the normal path to the way that they were doing it before, it turns out that they find a new way of doing it.
Yep.
And I think that ability to solve problems, to redeploy your genomically specified hardware in a novel way to achieve the same high level goal as best as you can under new conditions is just, well, first of all, it's just the tip of the iceberg. It's just what we happen to have noticed, right? I'm sure there's a ton more that these things. In fact, that's something I forgot to say. I meant to say this in my last question. I think it's very important to say that when you are trying to describe the intelligence of some system, we as scientists, when we look at something and we say, it has this level of intelligence or it doesn't, we ourselves are taking an IQ test, because it is very easy to underestimate the intelligence of something that you're not smart enough to recognize. And if you don't know the space in which it's solving problems, and if you're not very good at observing, just as one example, right? We are, humans and most animals are really good at noticing agency in behavior. All of our sense organs are pointed outwards. We see other animals running around and doing things, and we say, oh, look, it's burying something, it'll come back and get it later. That's pretty smart, or it's faking an injury or whatever. We're really good at recognizing that kind of intelligence. We are terrible at recognizing other kinds of intelligence because all of our training set, all of our organs point out, our sense organs point outwards. Now, imagine if you were born with a sort of a biofeedback sense, where at any point in time you could tell what was happening to your pancreas and what the pancreas did as a result of this, right? If you could feel that, if you could observe it the way we observe each other acting in the world, I have no doubt that we would consider that an incredibly intelligent agent that was solving problems all the time, right? It's just that we don't have that experience, so we're not good at noticing intelligence in other spaces. This is why we're still fighting about whether bacteria could really be solving problems in metabolic space, or if plants could really be intelligent. People have such a hard time believing that, because they're just not we're just not good yet at noticing intelligence in these other spaces. So I think the same thing is true here. We have to, this is just the tip of the iceberg in discovering what the novelty is, right? That these cells, and how we can, how we can take advantage of it.
From the computational side, I think there's a lot that's surprising about this particular study as well. Self-replicating machines also has a long history in Hollywood, and many sci-fi movies have treated it. You can trace it back to John von Neumann, one of the pioneers at the dawn of the computer age. And, von Neumann sat down to develop a formal theory of how kinematic self-replication might work. This was all theoretical in the 1960s. Von Neumann began by cutting his universe in half. There are machines, and then there are tapes. And a machine that has a tape inside, and if that tape has enough instructions to tell the machine, go find these pieces, put these pieces together, and oh, by the way, also make a copy of the tape and put it into this new machine, off you go, and you have kinematic replication. When Mike and Sam and Doug and I were working on this third paper, and we were starting to see kinematic self-replication in the dish, one of the things that I kept asking myself is, where's the tape? The machine, maybe it's a little more clear. It's the Xenobots, or maybe the piles, or maybe it's the cells. There were some good candidates for the machine, but where's the tape? In an obvious way, there's a tape. There's DNA inside each of these cells, but it's hard to argue that this DNA is directly driving this kinematic self-replication process, right? It's obvious that the frog DNA encodes frogs, right? As Mike was just mentioning, it's not so clear anymore. So I think one of the things that Xenobots are doing, and a lot of the biology that we're learning in general, is demolishing some of this dichotomous thinking that we have been using as a crutch for all these years. There is brain and there is body. That's Cartesian dualism. It's been with us forever. It hobbles our thinking. Genes are separate from phenotype. There's another clear distinction. And now, specifically in this experiment, it's a great, I think, exemplar of this concept. Where is the shape, and where's the tape, and where's the machine? We sort of tongue-in-cheek have been saying, the shape is the tape. The shape of the piles, the shape of the Xenobots is the tape somehow. It's dictating behavior. So I think, again, humans can only go so far with their intuition. And one of the first things we like to do is to cut the universe cleanly in half, right? Black, white, good, evil, brain, body. And all of that is being demolished by a lot of what's going on in biology and AI and robotics.
Very exciting. To continue just on these lines for a minute, just thinking about some of the things that you mentioned in terms of they're, specifically this case, they're, hey, all of a sudden they decided to explore this new state space. Obviously done the work early on with the self-healing. Are we learning anything yet from the Xenobots in terms of aging, longevity? Because I know, a lot of, I do a lot on this theme on the show, and as you always point to sort of some of the critters around this planet that are, verge on the sort of the biologic immortality domain, and they always have these interesting skills to not just reprogram themselves, but also shed off stuff that they don't want. Anything interesting yet? I mean, if you let the Xenobot just hang out and feed it and do its thing, how long does it go for? Could it go for years? I don't know if it's, forgive me if that's a silly question.
No, it's a great question, and I should say that sort of formal studies of aging are in the future. We've proposed a couple of projects on actually looking at senescence in Xenobots and looking at aging and what's going on there. But I'll tell you what we know right now.
Cool.
The standard issue Xenobot in plain water has a lifespan of about a week. They run, and what happens is they run out of energy. The energy that they get are little bits of lipid. So they're yolk platelets that are remaining in the cells that we take off of the embryo. They were originally provided by the mother. This amount of energy is enough to keep them running around for about a week's, let's say on the order of a week's time. And after that, they run out of energy and it biodegrades, and that's that, which is a nice, sort of safety feature and things like that. However, what you can do is, if you're very careful about putting certain nutrients into the medium, you can extend their lifespan. And it's a bit challenging because these nutritive media bacteria love to grow, fungus loves to grow. So you have to, it's kind of hard, and frog tissues aren't particularly clean ever. So it's kind of hard to keep these things, sterile. But if you do this, our record, I believe, is something like 85 days or so. You can keep it going. If you keep feeding it and cleaning it, you can keep it going. And I don't know, we don't know what the maximum is, because the 85 days, is I'm sure is not the limit. It's just kind of a practical thing. I bet we could get past that. During that process, they change shape. So they change shape. They become transparent. They have a very different shape. I think it's described in this latest paper, and or maybe even in the one before that. But so they have what you could call a developmental sequence. They change shape over time. They don't look like embryos. They don't look like frog embryos. They have a completely different developmental sequence. We can, again, have fun asking where that developmental sequence came from, if it wasn't specifically selected for, and what will happen to them if you can keep feeding them and keep cleaning them, and keep them going for a year. Or, frogs can live a couple of decades, 3 decades. What would happen to a bot over that period of time? I have no idea. This all remains in the future to be determined.
There's an old idea in science fiction that again, AI can help with immortality, right? We'll just eventually upload ourselves to the cloud. Couple of technical details. One of them is, what is this self that we're supposed to be uploading, right? Sure. And the answer is usually obvious. It's whatever's inside the skull. That's it. Just make sure that gets up to the cloud. But again, we're learning it's not that simple. So, and again, when you look at these Xenobots running around in the dish, as a non-biologist, some of these questions come up. Like, you have the first generation Xenobots that push cells into piles, which become the children, and they make piles, which are grandchildren. But are some of those cells just stray cells that are left over from the grandparents? Mike mentioned chimerobots, right? So sure. It isn't clear there's a distinction here between, who's old, who's young, who's generation 0, who's generation K, where is the Xenobot, where is the intelligence, residing in this system? As always, the answer is complicated. And sure. if the self is really something that can be subdivided and subsets of it can be propagated forward in time in very different forms, I think it opens up new ways of thinking about immortality, aside from the boring ideas of, uploading ourselves to the cloud. If we do, defeat death, it may be in some very non-obvious way, which is kind of fun to think about. Absolutely.
Guys, while I have you, we just entered 2022. What's hot on the Xenobot train for for the next 12 months, or stuff that's coming up, conferences we should be watching for you too? Please, both take the floor.
Well, from the biology perspective, one of the things that I'm most excited about, I mean, there's many things, too many to mention, but one of the things that I'm excited about is to really start to get a proper characterization of their baseline cognitive abilities. So yes, later on we'll add, more different kinds of machinery and neurons and who knows what else. But I would like to know, just out of the box, skin cells left to their own devices, rebooting their multicellularity into this form, what are they able to learn? What kind of learning can they do, if any? What are their behavioral preferences? What are their possible behavioral repertoires? Do they have things that they like? Are there ways to reward them? Are there ways to give them negative reinforcement? What's the IQ of that native construct? We're starting those experiments now, and I think I'm very interested in them as a model of basal cognition.
One of the other directions we're pursuing is just the pure engineering infrastructure underneath the Xenobots. So at the moment, we have automated design in the supercomputer. The evolutionary algorithm can churn out, billions or trillions of potential designs.
Mm-hmm.
As talented as Doug Blackiston, the microsurgeon, is, he can only turn out 1 millimeter-sized Xenobot every 4 hours or so. So we're looking to build some infrastructure where we can automate not just the design of Xenobots, but also the way they're manufactured. And that could be top-down or bottom-up. So it's going to be exciting in the not-too-distant future to see a Xenobot or some kind of biobot that rolls off the end of a very, very small conveyor belt. And it's the first quote-unquote organism that's been completely designed and manufactured all without human hands. I think that's going to be very exciting.
It is definitely going to be an exciting future, and it's going to be fun to continue to watch you two and of course Sam as well as Doug continuing on this path. I mean, obviously you've given us a lot to think about the last couple of years watching this work happen, and just really rooting your whole team on to continue what is clearly a very important area of scientific pursuit. For everybody that is going to be listening to this episode across the podcast networks or watching on our YouTube channel, you've been listening to Dr. Mike Levin, Tufts University professor, Vannevar Bush Endowed Chair in Biology, also director of the Tufts Center for Regenerative and Developmental Biology and director of the Allen Discovery Center. Also, Dr. Josh Bongard, professor, Morphology, Evolution and Cognition Laboratory, Department of Computer Science, College of Engineering and Mathematical Sciences, University of Vermont. We will also shout out to Dr. Sam Kriegman and Dr. Doug Blackiston, who didn't join us today, but have been instrumental in this work. Gentlemen, it was great seeing both of you again. I want to thank you for taking the time out of your respective schedules to do this. Thanks, obviously, for everything you've been doing. And as we say on this show, thanks for helping to create a better tomorrow via your research. Really, such a fascinating program.
Thank you so much. Yeah, really appreciate it.
My pleasure. Thank you.